Hello everyone. So today I'm going to be guiding you through how to determine the molecular weight size of a DNA band within a 1D SDS-PAGE gel. Now, obviously we use our Phoretix 1D software to be able to do this, and we've got lots of different options for working with commercial molecular weight ladders or your own molecular weight ladders and reporting those and things around that. So jump straight into it, start showing you how we add ladders to our gel and go from there. So the first thing we need to do is obviously we need to work through this workflow on the left hand side. So we need to define our lanes to be able to assign a ladder to a lane. I'm just going to use the automatic lane detection for now. That's not done a great job here because there's a lot of information missing between these different lanes. So I'm actually just going to, for speed, I'm just going to draw a lane box for these three here. And then I can come through and I can edit this because these lanes aren't really, there we go. It doesn't matter that I've left a little bit of space around my lanes here. The background removal will take care of that. So you don't need to crop either your bands or your lane boxes right up to your bands because the background subtraction will take care of that anyway if you use an appropriate setting, an appropriate background removal method. And you'll be able to see that in the next step, in fact. So if I come through here and show lane 2 might be a better example. So if I zoom out here, I'm just using my shift key and scrolling my mouse wheel here to zoom out. If I set rolling ball, is probably going to be a good one here. So we can just set this appropriately. So because I've not got show corrected on, what you're seeing here is the dark area underneath the peak here is what is actually going to be subtracted mathematically from my peak measurements. So we can bring that down as well, bring that down even further. So what I'm doing here is I'm just clicking and moving my mouse left and right, and you can see in real time how that affects the data. But what you can also do is, of course, you can do this manually, or you can type in numbers. So you can type in your own numbers here if you've got kind of an SOP on site that says you must always use 26, for example. And then if I turn on show corrected, you can see here is my 0 value. So my 0 intensity, and this is what my data is going to look like. So everything's pretty much removed apart from this peak, just as it was here. So this is kind of just a zoom in, zoom out type of situation. And I believe we can actually set the intercept. No, we can't set the intercept. It's just 0 is set there. So once I've done my background subtraction and I've applied that to all my lanes, I'm going to come down to my band detection. Now, when we run the band detection, we run it universally across all of our bands. So these settings apply to, well, by default, they apply to your molecular weight ladder lane and your bands of interest lanes. However, we do have the ability to only define different band detection settings per lane. So in a lot of instances, the strongest lane or the strongest bands in your lanes will be your molecular weight marker. And then all of your samples that you're looking to quantify will actually be weaker or lower in intensity than your molecular weight ladder. So what you can do is you can have two different sets of settings. You can have a set of settings which is fine-tuned to only detect very strong bands and use that on your marker lanes, and then separately use a different setup of parameters that are more tuned for faint band detection and use that on your samples. And then you can kind of remember those intensity settings, remember those automatic detection settings, and use them next time in further experiments. Now, actually, I can see here that the automatic band detection has done a pretty good job on the defaults. What I am going to do is just shift that up there so you can see here and shorten that lane. So what you can do for your molecular weight lanes is you can edit the bands. So you can edit where these bands start and stop. So you can edit your molecular weight ladder where that detection is occurring. This is really useful if when you apply your molecular weight ladder, it doesn't really apply where you want it to. This will make more sense when I run through the molecular weight ladder assignment. But there may be instances where you want it to skip a lane or you want it to there's a lane that was too close to another lane to be detected and you want to add it in manually, things like that. So you've got supreme control over where all of your bands are, but where all of your bands are for your molecular weight ladder lane as well. So I'm going to come down to my molecular weight ladder lane, and we can see obviously it's here on the left. Here's another example of a band I might want to go in and edit on my molecular weight ladder lane. We'll just bring that in here, make it nice and tight. I don't think I've got background subtraction actually on this lane. I think I only did it on, well, I think I've probably done it on all actually. So if I come through to my molecular weight settings, I can grab a number of commercially produced, typically used molecular weight ladders that we've got built into the software. Now, these are not maintained very strictly. So if you are using a commercial ladder, I would caution you to check that it is still relevant, that they've not changed the formula of the commercial ladder or the steps or the molecular weights of the commercial ladder since we included it in the software about a year ago. But assuming they are correct, what we can do here is we can just click the one that we've been using, left click, and then left click on our marker lane, and you can see it's going to start drawing these lines across now. This particular ladder that I've chosen has fewer steps, fewer markers than my actual lane does. So the software has automatically calculated the strongest, most intense peaks and used that kind of pool of steps to apply it to only the strongest within this lane. So it's tried to be a bit clever about this. It's tried to only target the most intense signals, thinking that everything else is noise if you're using a ladder that only has a few steps. If I come and grab one, I think this may be, I'll try and grab one that's got more steps in it. High range? Nope. Put the BioRad one. There we go, that's a little more appropriate. But you can see here that the software has automatically pulled out the peak of each of these bands and drawn your line across to perform your molecular weight measurement. Now, these bands are actually outside of our calibration curve for our molecular weight ladder, but the software will still mathematically extrapolate the values for those, even if they do lie outside of your molecular weight ladder. Now, a word of caution, if your bands do lie outside of the kind of reference values for your molecular weight ladder, it is less reliable, and I would strongly urge you to repeat your experiment with a ladder that includes the range that your bands are in. Now, the software will give you a result. We will mathematically extrapolate it, but it's a less accurate method of doing so. So it's important that you understand that. So I've applied my molecular weight ladder. So the software has all of the information it needs to kind of calculate distance migrated down the gel and associate that with a certain molecular weight, number of base pairs, kilodaltons of the protein, that kind of thing. And we can see on the right hand side here, when we assign a molecular weight ladder, the extrapolation that we do, we visualize that. So this is a standard kind of calibration curve where our brown points are our steps, and our brown point is our steps and our location, our migration through the gel, so our location down a lane. And then the green line is the mathematical line that we've drawn through them based on this curve fit type, from which all of your results will be extrapolated. So we've got a number of different curve fit types, and quadratic, exponential, linear, so we can support a great deal of different types of ladders. And we can also export all of these calibration curves. We can export them as an image, we can export them to a report in the final reporting step within the software. Do pretty much everything that you want with it. And if you want to add your own templates, it's as easy as adding a new template, giving it a name, so test 3, selecting the unit that represents your ladder. So if you're using base pairs, kilobase pairs, daltons, kilodaltons, percentage, pH, and then you simply select the number of steps that are in your ladder, and you manually enter the values to line up with the different steps that you have in your ladder. Once that's saved, it lives within your copy of the software forever, so you can keep coming back to it time and time again. Click save there. Now, once I've added this, I can go back to my band section and I can work out what my bands are. So what the extrapolated value for my bands are. So if I come down, if I turn on the band table, if I come down to lane 2, I've got one band in that lane and it's been detected at 0.8696 kilobases. That is because my one value is here and it's extrapolated downwards here. Oh no, sorry, that's the RF value. It's been calibrated as 0.9065. So now that I've applied my molecular weight ladder, you'll actually see that this unit has changed when I'm looking through my different bands. So if I just pop a few of these on here. So now instead of having position as the unit, so XY position within the image, we're actually able to look through our lanes and see where we can place the mouse, what that position equates to in kilobases based on this ladder. So we can start working with that information straight away for all of our lanes and bands. There's not many lanes and bands in this image, or certainly not many that I'm working on, but you'll be able to get a feel for how your different bands line up across all your different lanes, because when we're moving our mouse across here on the lane profile, it's synchronized. So you can see, for example, in 2 out of 3 of your lanes, you might have a band at exactly 2.6587, but in another lane, you may have had one that's shifted slightly. So you can work with it in real, well, not real world units, but kind of calibrated units rather than trying to link every intensity, every band to an XY position that doesn't really mean anything to you experimentally. So we've now got context for our experiment, not just results in this table. So we also have a number of different options that we can control once we, in the realm of molecular weight ladders. So if I come through to the settings menu here, we've got the ability to, as I just showed you, use the molecular weight scale on the x-axis. So we can turn that on or we can turn it off. So again, we're now back to pixel position. Again, is not super useful for someone within the experimental context. Turn that back on again. And we can also always show molecular weight scale on our image. So what that does is when we're working in our bands mode, we can turn that on or off to show our molecular weight band, to show our kind of ladder bands and where we're taking our measurements from. So I'll just turn that back on again. You'll be able to see this move in real time here. Settings. So you can see as I turn that on or off, can't really drop this around, but you can see the molecular weight ladder going on and off in the background. So when you've got it on, you can visually check the results that you're getting. So you can see where your bands line up with your molecular weight ladder to validate that you're using the correct extrapolation method, and that basically it looks right to you. So you can go back and double check the calculations that the software is performing to be able to do that molecular weight sizing for your bands. I think that's everything that we've got around the world of molecular weight sizing and calibration and quantification of unknown DNA bands within the software for now. Of course, it's always liable to change. We're constantly improving, constantly updating our Phoretix 1D software. So if you want to try out a copy of the software in your own lab with your own images, please check out the link in the description below and leave any feedback in the comments, or contact us through the website about if there's something missing from the software that you'd like to see, or how you've got on sizing your DNA gel bands and working out the molecular weight for them.